much. Can you hear me okay?
Yes.
Okay, good. So title of the presentation is Organic Carbon Retention in Natural and Urban Soils as Affected by Land Use and Management. And, okay, short general introduction. We already learned a lot of about soil organic matter, just a little review. So key parameter for functioning of soils, because it's important for water storage and water quality. It is important for nutrient storage and supply. It is important for structure and aggregates acting as a glue. It brings down the density, the bulk density. It is decreasing erosion by stabilizing the soil structure. We store greenhouse gases in soil organic matter, right? Therefore, we have the soil as a storage for greenhouse gases and mitigation of climate change, habitat and productivity, and it is coupled to biogeochemical cycles of nitrogen. So in soils, we store more carbon than in the above ground biomass and in the atmosphere together, right? So just to give you a perspective. So that are the controls. So we have soil mineral characteristics. Clay content is one part, but also cations, iron oxides, aluminium oxides. So mineral, inorganic, mineral characteristics are responsible for stabilization. And if I'm talking about stabilization, that means protection against microbial decomposition. Then we have soil and land use, which is arable, forest, grassland, but also urbanization. So urbanization is a land use that grows with the biggest speed right now, right? Soil management, so what we do, tillage, cover cropping, biochar, and so on, but also soil depth. So we are touching, with my data or what I presented in the results, we are touching a little bit of everything here. So what we see over the last 2,000 years, we have an increase in land use intensity, which goes hand in hand with a loss in soil organic carbon. So if we change a natural system into an arable or a use system, we are losing soil organic carbon, and that led to an average reduce worldwide topsoil for 30 centimeter down by 26%. That goes up to 80% in certain sites, right? But we're also losing 60% for the top 100 meter, right? And I will show you a couple of data. We have data that goes down to 3 meters deep. So the question is how we do get this carbon back, right? That we lost over the last 2,000 years, but especially over the last couple of 100 years of intensifying our land use. Okay, challenge, better understanding of factors, processes, and mechanisms that drive soil depth specific differences in carbon storage, stabilization, and decomposition, because this is critical for soil management decisions, land use strategies, modeling efforts, and terrestrial carbon budget. That is the first case study I would like to talk about. It's the storage and turnover of organic carbon in 3 meter soil depth. Insights from 22 arable and native prairie sites in Nebraska. Study was led by my former PhD student, now a doctor, Chris Newell. One paper is out in Plant and Soil. The other one is under major revision with Geoderma. So Chris was leading this effort. And what we did was we sampled in total 22 sites in Nebraska down to 3 meters, and then we subdivided, so per site 3 cores down to 3 meters, and we subdivided this cores into pedogenetical horizons, so A, B, and C. And this were our sites over here, so we go here, especially in southeast Nebraska. One plot was in Holden. That's the oldest field experiment we have in Nebraska. It's over 100 years old. And then we sampled the soils, and analyzed them for bulk density, pH, texture, but also for cations, aluminum and iron oxides, carbon isotopes, microbial community structure, but we also used the fractionation method. And we had a couple of talks already touching this, right? We have the bulk soil organic matter, we can subdivide it in ecological meaningful fractions. So the soil organic matter, particulate organic matter, mineral-associated organic matter. These different fractions are turning over differently, therefore they have a different function. As more quicker they turn over, as bigger their importance is for microbial substrate, as lower their turnover with a higher turnover time, as more important they are for the long-term storage of organic carbon in soil and the function of soil for mitigating climate change. So that was our, so we looked at fixed depth increments and then our horizons. So you see our horizons, they are pretty heterogeneous. So the A horizon can go to 12 centimeters, but also to 38 centimeters. The B horizon can start at 12 and can go with until 180 centimeters. The C horizon can already start at 28 centimeters and goes down to 300 centimeters. And this A, B, and C horizon, these are kind of biogeochemical boundary conditions. So these horizons are there for a reason, because we have changing conditions: organic matter content, clay content, parent material, and so on. So these are a range in soil characteristics that we have in our soils, and these soil characteristics are important for carbon stabilization. So we have a pretty good range in clay content, which is important, so 13 to 30 here for the arable, in the A horizon, but also aluminum oxides, iron oxides, calcium, exchangeable calcium ions. So calcium ions very important for stabilizing soil structure and aggregates. So we have a good range in these parameters that should reflect carbon stabilization and carbon storage. That is the bulk soil carbon storage in kilogram per square meter. If you want to convert this in tons per hectare, you take it times 10. That is fixed depth increment, right? So we went from 0 to 50, 50 to 100, 100 to 200, 200 to 300 centimeters. And you see the biggest difference, significant difference in carbon stock is in the first 0 to 50 centimeters. So here, by a change of arable land into, from grassland into arable land, the majority of carbon we lose is in the 0 to 50 centimeters. So everything else, we didn't see differences between this arable soils and this native prairie soils. So this is according to A, B, and C horizons. That's the same story. We see the differences in the A horizon, but not in the B horizon and in the C horizon. But about 70% across biomes of organic carbon is stored below 30 centimeters. So your subsoil compartment for carbon storage, carbon sequestration is quantitatively highly important. So we have about 70, give or take, percentage stored of organic, soil organic carbon in the subsoil. So then we did some isotopic analysis. Here you see the delta 13C. Delta 13C again, A horizon, B horizon, and C horizon. We don't see differences between the land use systems, but we see a depth influence. So the delta 13C gives you a bit of information about the origin of your carbon material. The negative 15, that gives an isotopic signature of C4 plants. A C4 plant is, for example, corn. There has a different photosynthesis that results in different isotopic signatures. And this comes from introduction of corn, but also C4 grasses in the prairie. In the C horizon, you have a delta 13C content that is indicative for C3 plants. And this stuff grew on the native prairies after the glaciers removed. So, and if you look at the 14C data, this is over here. So 14C is a measurement to see how much old carbon you have in the soil. And to give you a number, the C horizon, we don't see differences. That's about 12,000 years old. that is from Ice Age. Right? That is in the soil since the glaciers retreated. So no differences. So we know there's carbon going down, but the microbes decompose it immediately. But the old stuff is highly stabilized and stays. The bio horizon doesn't show difference. It's about 5,000 to 6,000 years old, but we see a land use impact in the A horizon. So the grassland is about 200 years old. The arable stuff is about 1,200 years old. So what happens, or our explanation is, if you change grassland into arable, the younger material is less protected and is decomposed preferentially, leaves the old material behind. So you have an enrichment on old material by increased decomposition if you disturb a grassland ecosystem. What we also did, we did like microbial community structure with DNA analytics. I'm not a microbiologist, so I will keep it down or to the basics. What we see is in the A horizon, that is total DNA that gives you a proxy for microbial biomass. That is 3 times more in the grassland ecosystems compared to the arable systems. So if you disturb your system, you bring down the microbes, right? That is, all this is fungi and bacteria, microbial community structure. So we didn't see that much differences, a few, but here we need help from real specialized microbial biologists. I'm not, so we didn't find that much differences, but if you probably go more into the metagenomics, you might see some differences. So I will keep it here that way that we see the decrease in microbial biomass. So what we did then, so that's take-home message. So overall, the study indicated that most soil organic carbon gains and losses and transformation and translocation happens in the A horizon or uppermost 50 centimeters, and the carbon isotopes indicated that land use change affected SOC persistence in the topsoil, but not in the subsoil horizon. So and then we talked a little bit about influence of management, land use on these different carbon fractions. So that is a hot topic. So there are a couple of papers published over the last years in Nature and PNAS, and so highly published papers and highly impact journals. So what we did, we took the soil samples. So I'm fractionating organic matter fractions since over 20 years. Our group developed over the last 2 decades a method that allows you to separate sequentially free particulate organic matter, which are organic particles or root litter. Free means it's not aggregate occluded, so it is highly relevant for substrate for microorganisms. Water extractable organic matter is highly important for transport of energy and nutrients in the soil and organic carbon, but also as a substrate. Then you have aggregate occluded particulate organic matter, which is highly important for structure and aggregate stabilization. And then you have the mineral associated organic matter. And over here you see the mean residence times, right? So free POM between 1 and 50 years. So there's always site depending and environmental condition depending a variability in mean residence time. And if I'm talking about a mean residence time of 50 years, that is, the concept says it takes 50 years to exchange each carbon atom at least once. Right? So as higher the mean residence time, as longer is the pool in the soil, and as higher is the organic matter stabilized. So the long-term stabilization, especially mineral-associated organic matter, because this protects the organic matter from decomposition. I will show you data that actually says goes in the other direction, at least a little bit. So FBOM, water extractable organic matter, more important for substrate and microbial biomass and decomposition processes as a substrate. OPOM and mineral associated organic matter, more important for long-term carbon stabilization. So these are data from our soil organic matter fraction, so it's pretty busy. Here is the FPOM, here is the OPOM, that is water extractable organic matter, that's mineral associated organic matter. Then we have our A, B, and C horizons. And if you look at the black numbers on top, you see the mineral associated organic matter more than 50% of total carbon. So it is quantitative the most important organic matter fraction. Particulate matter, water extractable 10% or less. Land use effect, we see in the A horizon for each of the fractions a land use effect, so bigger in grassland than in arable. Interestingly, water extractable organic matter in the C horizon for over here and in this red circle, so higher in the subsoil for grassland ecosystems. So it shows you that the water extractable organic matter is highly important for deep soil carbon dynamics and also as a diagnostic fraction. Interestingly, the mineral associated organic matter, which is actually stabilized against, or should be stabilized against decomposition, is lower in the arable soils than in the grassland soils. So that shows you that a part of this mineral associated organic matter gets decomposed by land use change. And what we also have seen, that is the CN ratio. Just look at the CN ratio of the mineral associated organic matter. That goes down as well. So in our explanation, so we already seen this, that is the famous onion layer model developed from Markus Kleber and co-workers. And what it says is that this contact zone, that is the highest stabilization. The hydrophobic zone and the kinetic zone, that are zones that are more closely into the water dissolved phase, and they are more easily accessible for microorganisms. So microorganisms attack first this zone, then that zone, and if you see over here, a lot of proteins, right? Nitrogen containing compounds in a smectite environment. In our Nebraskan soils, we have a lot of smectite. So our explanation is that if you turn grassland into arable, mineral associated organic matter is in part decomposable because they decompose the two layers, and if they don't have access to the nitrogen bonded layer directly protected with the mineral surfaces, then your CN ratio goes down. And this is what we observe in our mineral associated organic matter. All right, then we looked into relationships between the different fractions and total soil organic carbon. So here, again, busy A, B, and C horizon, FPOM, macro-aggregate occluded POM, and water extractable organic matter. And here you have the soil organic carbon. So what you see, the relationships here and there, you have a correlation between the fraction and the soil organic carbon, but keep in mind the fractions are below 10%. And if you look at the mineral associated organic matter, independent from land use and your horizon, the mineral associated organic matter is always strongly positively correlated with your soil organic carbon content. So that's interesting because this drives, so that's a back and forth relationship between bulk soil organic carbon and mineral associated organic carbon. So now it gets, so here we try to correlate this mineral associated organic matter and bulk soil organic matter with our mineral characteristics. That are the arable soils, native prairie soils, A horizon, B horizon, C horizon. That makes sense. So we have native prairie, so positive correlationships between mineral associated organic matter and soil organic carbon. And we have the positive relationship with clay, calcium, magnesium, aluminum oxides, and so on. This is a bit overlaid by management effects. So here, it matters if you have cover cropping or irrigation or tillage, but that, I need to tell you, we're still struggling explaining this data because this shows you a different direction, right? Higher clay content, lower soil organic carbon. Higher calcium, lower soil organic carbon. Right now, our best explanation is very heterogeneous and substrate limited environment. Clay lenses or clay is also always attracting water and nutrients. So maybe they support microbial decomposition processes. We don't know. And in the sea horizon, it looks again as it should look like, but our take home here, correlation patterns pointing towards a complex dynamic in factors controlling organic matter mineral interactions across pedogenetic horizons. So, losses in the mineral-associated organic matter accounted for about 48% of losses in the bulk soil organic carbon. The mineral-associated organic matter in this high-input environment, horizon prairie, is substantial proportion prone to losses. But at the same time, this mineral-associated organic matter is kind of our last frontier, right? It is 60% stabilized, highly stabilized against disturbance. So that is stuff that is highly stabilized against microbial decomposition and stays in the soil. And findings highlight the relevance of mineral-associated organic matter for soil management and land use strategies that aim to restore soil organic carbon to pre-disturbance levels. So that is the second case study I would like to present. I was led by Dr. Lidong Li. She was a former postdoc in my lab. And it's about consequences of conversion of grassland to coniferous forest for topsoil and subsoil carbon dynamics. So in the Great Plains over here, we have this problem of red cedar invasion, so grassland encroachment. So that is actually threatening the backbone of the beef production in the Great Plains, because that goes pretty quickly that the red cedar transforms a grassland into a forest, right? And if you want to graze cattle, you don't want to have a forest on your rangeland. So, and that is an area so-called the Sandhills grassland of Nebraska. So that's a unique ecosystem. It's 14 million acres of the most intact prairie ecoregions in the world, and it's the largest stabilized sand dune formation in the Western Hemisphere. So here we were, so in general, if you go temperate grasslands and shrublands and convert this into temperate forest, you're losing carbon, right? It goes in the total from 300 to 160 because you're losing a lot of carbon in your soil, right? So here we have large uncertainties remain in understanding of how woody grassland encroachment affects carbon storage and persistence beyond the topsoil. And we wanted to look into the drivers of the subsoil dynamics. Objectives were, again, looking carbon storage in the uppermost 3 meters, looking into carbon turnover dynamics, and looking into microbial biomass differences. That is a study site. So we had a native grassland over here, and we had 2 different coniferous forest ecosystems. Again, we sampled deep to 3 meters. We didn't see really pedogenetic horizons, so we sampled in fixed depth increments. The sand ecosystem really do not form. It's only an A and a C horizon. The A horizon was 10 centimeters, and then the C horizon started, so we sampled in fixed depth increments. So land use, red cedar, ponderosa pine, and native grassland. The forests were 64 years old and 82 years old. That were Nebraska forest experimental station. So they planted it on purpose to see how it changed over time. The soil is more than 90% sand. So that is a unique soil system, right? Almost no clay, no calcium, and so on. It's just sand, almost pure sand. And then we looked again in carbon stocks, delta 13C, mean residence time, and microbial biomass. So if you look at the carbon stocks, in 0 to 10 centimeters, you could think if you transform a grassland ecosystem into a forest, you gain carbon. But if you look deeper down, it looks different. Right? In the subsoil, you have much more carbon. We have more carbon in the grassland ecosystem than in the forest ecosystem. So and if you look over here, 0 to 300, 100% of calm, and again, here we have in below 30 centimeters, 50 to 70% of this calm, even in this high sandy environment, right? And because of this effect on the subsoil, you have in total, in 0 to 3 meters, more calm storage in the grassland as compared to the forest. But if you would just stick to 0 to 10 or 0 to 30 centimeters, you wouldn't see these differences, right? You could conclude, okay, I should change my grassland into forest so I store carbon. But the opposite is the case, right? Under organic carbon considerations, you should do everything to keep your grassland. Beside that is in rangeland and in usage, right, by the farming community. So that is mean residence time, so we could calculate this a little bit based on changes in data 13c. Not really surprising, topsoil turns over more quickly than the subsoil. Interestingly, between 10 and 300 centimeters, we don't see difference in the mean residence time. It is not significantly different, and the sandy soil, there's a really homogeneous resource distribution, water, nutrients, and so on. So that means that we have almost always the same conditions for microbial organic matter turnover. Therefore, we don't see really much differences in the stabilization or destabilization. So, but in general, the pine forest organic carbon turns over a little bit more quickly than the cedar forest. Topsoil turns over more quickly. It's also not surprising. Surprising is really that we don't have really differences in the subsoil environment. So and then we looked a little bit more in detail into the microbial biomass. So we measured here with the FAME, that's fatty acid methyl ester. And here we have seen that especially in the subsoil, we see more microbial biomass in the grassland. And there's a little bit of a paradigm shift over the last 10 years. So meanwhile, it is estimated that 50% of soil organic carbon comes from microbial biomass or microbially processed plant-derived organic matter. So our explanation is that it cannot explain everything, but it can explain a little bit or a part. That the grassland, that the forest reduced the microbial biomass in the topsoil. Therefore, we have reduced carbon in the no, the other way around, I'm sorry. So we have a higher microbial biomass in the subsoil from the grassland. So that would at least explain in part why we see a higher microbial or total organic carbon storage in the subsoil under the grassland. So and that makes sense because there are two review studies showing that grassland, that is microbial biomass and fungal biomass, contributes much more to the soil organic carbon as compared to forest, right? So in grassland, microbial biomass is much more important for the total organic carbon than under forest soils. And our take-home message is, so conversion of native grassland to cedar or pine forest increased stock in topsoil but decreased SOC stocks in subsoil. The total soil organic carbon stock is higher in grassland than in forest. That goes for the 0 to 3 meters. Then we have 75 to 70% in the subsoil, 30 to 300 centimeters. And the decrease in SOC storage was associated with reduced microbial biomass and altered microbial community structure under forest relative to grassland. So the decrease in microbial biomass under forest might be mediated by declines in soil moisture, pH, and nutrients. So we are not sure right now what are the underlying mechanisms, but we are pretty sure that the shift in microbial biomass because of land use shift was responsible or in part responsible for the different soil organic carbon patterns. Okay, coming to organic carbon storage in urban soils. There are 3 papers I want to highlight because we don't have that many data in our group on urban soils. So first one is from Sheen et al., published in 2022, and they subdivided carbon storage into natural soils, urban green space, and urban intensive. So with urban soils, it's a little bit tricky because in an urban area you can have native soils. But you can in urban areas you also have urban soils. The urban soils are characterized by a certain degree of anthropogenic influence. Extremely high bulk density, a lot of artifacts, bricks, debris, high pH, low pH, contaminants, and so on. So here as an example, Central Park in Manhattan, New York was defined as urban green space, while roadside soils in the commercial areas of lower Manhattan were classified as urban intensive. So here are some pictures for the native soils, the urban green space and urban intensive soils. And you see that the native soils as tons per hectare have a little bit more than urban green space and urban intensive. But if you put this into perspective and you calculate in the entire area globally that is covered by these different land uses, you see that this urban green space, that's a paper by Guo et al. They said, so they found basically the same amount, 50 tons per hectare, but it's only 0.3% in the uppermost 20 centimeters globally, because this urban green space covers less land than our grassland or forest ecosystems. And then there was a nice paper from UC Merced, Malone et al. They looked into different urban soil classification. So it was restoration, landscape, gardening, and recreation. And looked into different amendments, so that one is control, compost, biochar, biosolids, and a combination. And you see over here that if you have the control soil and an amended soil, you increase your soil organic matter. And then they looked into different parameters with this different amendment types. So soil organic matter, again, biochar has the highest impact. Under phosphorus, you see the biosolids. Biosolids, a lot of nutrients, high in organic matter, high in water. And then we look into potassium, pH, and bulk density. So here we have kind of alternating effects of biosolids and and compounds. And because I talk to a lot of people here, we also have, or in UNL, my group is also working on biochar. So Dr. Blankenship already mentioned this, right? Biochar is still very expensive, but biochar is the only option right now that we have to increase our organic carbon content significantly, site independent, immediately and in the long term. So more than 90% are stable for more than 100 years. If you apply manure, compost, or biosolids, in 10 years, 90% is decomposed. 10% stays behind. So compost, manure, and so on needs to be applied every year or every other year to see an increase in carbon. Also, carbon storage gained via cover cropping. As soon as you disturb the soil, you're losing your carbon again, right? That happens not with biochar. And biochar is also very important because it has a couple of co-benefits. So it can increase water retention, it can increase nitrate retention, it can increase nutrient retention, it reduces N2O emissions, it can be beneficial for clay soils, improving water infiltration. It's highly efficient for coarse soils and sandy soils. Here we see the biggest effect on yield. Despite biochar is not a fertilizer, it's a soil conditioner. And we have 3 field experiments in Lincoln. Okay. that is a field experiment where we put 6 years of cover crops and then we split our plots and applied biochar. We went here a little bit crazy. We applied 70 tons per hectare. But we increased our carbon content by this application, one-time application, so that's a once-in-a-generation investment, by 60 tons per hectare, immediately, up to 0. I need to tell you, we had wind erosion, so we lost a little bit, but our highest increase was 60 tons per hectare, right? So, hectare, 60 tons per hectare, so it's 30 or 25 per acre? It should be divided by 2.5. Yeah. So that gives you a number how much you can increase, right? If you do it the right way. But field application is always, different. So our lowest was plus 30 because we lost via wind erosion, or during application it was a windy day. I mean, we have every day is a windy day in Nebraska, but this day was especially windy. And we lost a little bit where we don't know, but if you measure under field conditions, it's always, you have your landscape variability, your field variability. We need to pin down the numbers again because we are measuring now every year, but that works. We're also looking in microbial community structure, in carbon fractions, in greenhouse gas emissions, so we're trying a more holistic approach. And what we also did, that was last year, so this is among the biggest biochar experiments in the US, I think. It's 16 acres. We applied together with biosolids or alone, so biosolids, biosolids plus biochar, and the control. The idea was here to reduce the amount that you need to increase your carbon content. So we applied in total over 120 tons of biochar on this site. So 16 acres, each plot is 1 acre. So that was started last year. So the biochar is supported by Nebraska Forest Service, Energy Center, UNL, and a hedge project. So, and because I could talk another hour about biochar. It's just, starting with the Terra Preta and the kiln sites in Germany, we have this long-term. So I always love to tell my students about the Terra Preta soils that are soils in the Amazon, 1,000 years old. Indigenous tribes, they put biochar, charcoal, biosolids, food residues in their soils, and we still see this Terra Preta soils 1,000 years later. And the soils in the Amazon are poor, that are oxysoils. Low pH, no organic carbon, and 1,000 years later you still see the Terra Preta soils. Right? The old kiln sites in Germany or in Europe where they produce biochar for energy purposes, you still see this in a field 200 years old, you still see the spots, you still see a higher carbon content. So we have this long-term field experiment that shows that this is working. And we are organizing a biochar conference in Lincoln, end of September. I would be very happy to welcome you. We will share the link for the homepage. So if you have time, just join us in Lincoln. We try to bring together all stakeholders, so production side, policy side, education, research, feedstock, all in the biochar business. So we're trying to bring together and have 2 or 3 days discussion on biochar. What we also do is the Nebraska Urban Soil Initiative that is led by Dr. Wardman, which is a colleague of mine, mainly funded by NRCS. And that is a citizen science project. I'm a co-PI here. So we have about 400 active participants, and they committed 100 square feet of their garden to implement a new soil health management practice and collect soil samples 3 times. And they are asked, or they can choose between cover crop, no-till, compost, biochar of course, and some mixture. So cover crop plus no-till, cover crop, no-till, compost, cover crop, no-till, compost, and biochar. So that is up to the participants, what they use. What we want to do is, Christian Stevenson, that's another colleague of mine, also a co-PI, he did a little bit of survey and looked into motivation and pre-knowledge, and you see most participants had a positive, very positive, or somewhat positive perception about this certain soil health measures. That was probably the reason why they participated, right? Because they are interested in this kind of stuff. And this was a little bit motivation. So to try to learn something new, improve soil biology quality, so microbiome, microbial biomass, it's always a hot topic. Participate in research, improve soil structure, increase soil organic matter. So you see there's a certain amount of knowledge already, right? That the gardeners who are interested do have, so they care about their systems, right? They think about their system. All right, so soil analysis. Participants collect 4 to 6 cups of soil to a depth of 6 inches randomly, and then they send it to us at 0.0, 12, and 24 months after incorporation or implementation of the soil health measure. So first samples at 0 months, 317, then 117 at 12 months. So it goes always a little bit down, but that's fine. I mean, 170 samples is totally cool. And then we look after 24 months, 0 and 24 months, we also want to do a calm fractionation. In this case, we go for POM, particularly in organic matter, and different aggregates. So bigger aggregates, smaller aggregates, very smaller aggregates. So usually your turnover time goes down, right? No, goes up. So that one is mostly stabilized, and this one is less stabilized. All of them is important for stable soil structure. All right, some data, ground zero or time zero. Soil pH is a little bit above 7. So perfect for most nutrient availability is 6 to 7. So it's a little bit too high in average, but still okay. So circumneutral is fine. That is soil organic matter, on average around 6. So we want to see if we can increase soil organic matter content with our soil health measures over 1 year and 2 years. Organic carbon content, CNN is pretty low. So as lower your CN ratio, as higher the decomposability is, but as better is your nutrient supply from your organic matter. And last slide, if you really want to go and analyze urban soil and carbon storage, that can get tough. Because you can get situations like this one. Right? So if you have a truly urban soil with 200 years or 100 years of anthropogenic influence, you change the battlefield, right? You have high pH, low pH, you have contaminants, all of this stuff is bad for microorganisms. And usually organic matter goes into CO2 because this is thermodynamically favorable. But there are restrictions to this full metabolic potential of microorganisms. And soil organic matter needs to be considered through the lens of microorganisms. So what helps them to decompose it? Water, nutrients, temperature. What prevents them from decomposing organic matter? Mineral associations, aggregate stabilization. Freezing, what we see in the permafrost region is a change in environmental conditions that favors microbial decomposition. The permafrost is thawing. As soon as the microbes can take over, they decompose stuff that's 30,000 years old because it was frozen, right? You change your environmental conditions and you bring down or bring up microbial decomposition. In urban soils, because we have native soils in an urban environment, we have highly anthropogenic altered soils in an urban environment. So carbon storage and carbon stabilization in true anthropogenic urban soils gets complicated because you have this contaminant, right, that might even prevent any microbial life. I mean, that's not really possible, but most of it, right? or if you have really low pH or really high pH value. So that all can, and if you have, you know what they did in my backyard, they removed the topsoil and now I try to grow grass on the subsoil, which is 80 or 120% clay, I don't know. But in this case, I'm introducing grass or plants and I'm putting calm in the subsoil where no calm was before. But if you have a soil that is full with all calm and then I put a concrete on it, right? That is sealing it off. So I store my calm forever. The colleague of mine is mixing biochar with concrete. That is the ultimate calm sequestration. That stays there. I don't know forever. If you have biochar and concrete and build a house or a city, so you can be pretty sure you have a good calm sequestration.
Yeah.
All right. Thank you very much.